Good morning everybody. Today we are supposed to discuss about the various type of microbial interactions which are going on in the soil. Now here the question arises that why the microbial interactions are going on or why they are essential. Now here let us focus that the evolution of life is mainly depending upon the various type of interactions which are going on in the soil. They are necessary for the evolution of life, for the development of some new type of characteristics. They are also responsible for the ecological stability. They allows the organism to utilize the nutrients which are present in the soil very efficiently, or you can say that these microorganisms acquire the capacity or they acquire the ability to use these nutrient resources. Now we are discussing this topic into three broad categories, particularly in terms of introduction, then again regarding the microbial flora of soil and various type of microbial interactions which are going on in the soil. Now again in the introduction, let us look that the interactions which are going on in the soil. What is this soil or how it is being made up of? Soil is particularly that fertile region of earth which is providing all the vital nutrients which are essential for the growth of living organisms. Now how it is being composed or what are the constituents which are allowing these type of interactions? Now it contains different type of minerals, the organic residues, it contains the gases, water and large number of microorganisms. Regarding the minerals, there are two categories, the dominant minerals and less dominant minerals. The dominant minerals particularly the aluminium, ferrous and silica, whereas the less dominant one includes the sodium, potassium, calcium, titanium, phosphorus, nitrogen, etc. are present in the soil. Whereas regarding the organic residues, the dead and decomposed animals which are present inside the soil or on the surface of the soil, they are undergoing for the degradation and they are making the soil. They are again responsible for the formation of humus, and this humus ultimately increases or improves the water holding capacity, the buffering capacity and the aeration capacity of the soil. Regarding the gases, carbon dioxide, nitrogen and oxygen are some of the major gases which are present in the soil. Regarding the water, the climatic environmental changes and the drainage are the major sources. And what is the role of water? Water provides all the essential nutrients in the solubilized form which can be easily utilized by a large number of these organisms. See, here we are using the term organisms. We are not talking about the microorganisms at present. Now we are moving towards the microorganisms. That what are the living organisms? First of all, living organisms includes large number of trees, their root system, and this root system is having the rhizosphere which is holding tremendous number of bacteria, actinomycetes, algae, fungi, protozoa and different viruses. Now again regarding the flora or the microbial flora of soil, it contains the number of bacteria which ranges from 1.5 million to 250 billions. Regarding the actinomycetes, it ranges up to the 7 lakh number, whereas regarding the fungi, the number is up to 4 lakh. Regarding the protozoa, in terms of 40,000, and you can say that this large number per gram of soil has been achieved from the research journal, and it indicates that fertile soil will always be holding this large number of microorganisms. Ultimately, this large number of microorganisms, their activities, their interaction between each other is responsible for various type of interactions which are going on in the soil. Now let us look that what are these interactions. But before going for that, we can say that there are some of the abiotic factors as well as some of the biotic factors which are playing an important role. Regarding the abiotic factors, you can say that the availability of the nutrients, aeration, moisture, etc., or the temperature or the pH, they are playing an important role for the upliftment and the growth of microorganisms. Whereas regarding the biotic factors, the same thing, that large number and the types of microorganisms are responsible for these interactions. Now let us look for different type of microbial interactions. This has been typified into three categories, that neutral associations, positive associations and negative associations. The neutral association indicates towards the neutralism. We used to call it as neutralism. Positive interactions are being categorized as a mutualism and commensalism, whereas the negative interactions are being classified as antagonism, competition, parasitism and predation. Now let us first look for the neutralism. The word itself indicates that it is a neutral association. It is not going for any type of interaction, or you can say it is a lack of interaction between different organisms. Now ultimately it itself indicates that when there is a lack of interaction, then these organisms must be existing at a very low microbial population. Again, they must be existing in the adverse environmental conditions. They must be existing in the resting form, in the form of cyst, or in the form of spores. Now these are the neutralism type of associations. Now let us look over our major aspect regarding the positive associations. Positive association mainly regarding the mutualism. Mutualism is an association between one or more than two organisms, where each organism is benefited, but neither of the organism is having any harm over its growth. Again, we can explain this mutualism in terms of synergism, syntrophism, mutualistic symbiosis, and endosymbiotic interactions. Now what about the synergism? Synergism is an association between two organisms where both the organisms, they are providing the essential nutrients to each other. They are producing some of the extracellular enzymes which are undergoing for the degradation of the insoluble materials like lignin and cellulose which are existing in the soil, and they are particularly made available to the organisms. Now this synergism, one of the very important thing is the association is not necessary that one organism may be replaced by any other organism, and in that way it is being classified as a different category. Now let us look for the another one, that is syntrophism. Now syntrophism is the association between two or more than two organisms which are living together, two or more microbial populations which are living together. And again here the important thing is they are individual organisms, they are unable to utilize the nutrients, where each organism's involvement becomes essential. We can quote this with one of the typical example, that as for example arginine is supposed to be utilized, then arginine is not utilized even by the Enterobacter faecalis, Enterococcus faecalis, or E. coli, both the organisms, they are unable to utilize. But if it is being degraded partially, then Enterococcus faecalis will be utilizing this arginine into ornithine, and now this ornithine will be utilized by E. coli into the putrescine. Now this putrescine is being utilized by both the organisms, Enterococcus faecalis as well as E. coli. In that way we can say that putrescine is made available to both the group of organisms. So syntrophism is again one of the association where more than two microbial populations may be involved, and their cumulative effect give you the product which can be utilized by individual member of the different microbial populations. Now let us look for the another one, that is mutualistic symbiosis or symbiotic mutualism. Now this is one of the obligatory association. Both the partners, they are seeking the benefit from each other, and once this association is going on, then both the partners must produce some of the useful substances towards each other. Here some of the very common examples in terms of the nitrogen fixation, where the nitrogen fixing bacteria, they are going in association with the roots of some of the leguminous plants. Bacteria, they are living inside the nodules, they are fixing the nitrogen which is made available to the plants, whereas the plants ultimately provides the nutrients which are essential for the growth of this bacteria. You can say that the bacteria like the Rhizobium group, Rhizobium meliloti, then Rhizobium loti, and Rhizobium japonicum, they are particularly involved in these type of interactions. Now again the another very common example is regarding the lichens. Now what are these lichens? These lichens are the cumulative association together growth of, giving rise to a new growth of organism that we used to call it as a lichens. It is the growth of algae along with fungi. Both are growing together, and they are growing on the substrates where the green plants cannot grow. Here the fungi is providing the nutrients and moisture to the cumulative organism, whereas the algae ultimately fixing the carbon dioxide, producing the starchy material which is being made available to the growth of the complete fungi or for the growth of complete lichens. Now here once again look that these lichens, they are very important with the interaction point of view, or they are important with the humankind, that human beings obtaining various types of dyes and colors from these lichens, different type of perfumes, they are also being prepared from these lichens. And again these lichens, their growth is one of the indication of pollution, that if the pollution amount is increased, that these lichens they are unable to grow. Now friends, here let us look that these lichens they are growing at a higher altitudes, they are also growing at a low temperature. You can say that they are the indicators of different environmental changes. And one of the very common example that we are using in our routine laboratory regarding the litmus dye, that is also being acquired from these lichens. Now one of the another example regarding the mycorrhiza, this is the association which is existing between the green big plants and the different fungi which are living in the soil, particularly the region nearby the periphery of the roots, on the surface of the roots, outside the roots, and they are undergoing for the interaction with the roots. These mycorrhizas, they are again being divided on the same basis that where these fungi are growing. They are divided into two categories, that is the ectomycorrhiza and the another one is the endomycorrhiza. The roots of the plants, they are covered by the growth of this hyphae. The mental hyphae is formed over the surface of roots. This mental hyphae helps the plant to absorb the maximum moisture and nutrients from the surrounding environment and make it available to the plants. The Pinus sylvestris is one of the best example of this type of ectomycorrhiza, where this plant is being inhabited by more than 40 different types of fungi. Any one species may go for interaction at a time, or you can say that even more than one fungi may go for interacting with these plants. The another association is the ectomycorrhiza, where these organisms, endomycorrhiza, again the fungi, they are penetrating inside the cells, they are going inside the tissues, and the same work, the absorption of the nutrients, providing the moisture, directly being made available to the plants. Plants in turn, they provide the secreted organic substances as well as the nutrients. So friends, here you can see that without the interaction or without the association, the survival is not possible. So this might be one of the natural phenomena that these organisms, they are acquiring one of their ability to go for a symbiosis, to go for the association. Now these fungi, adapting to the environment, they have developed themselves for making some of the specific characteristic organs like arbuscles and vesicles. These arbuscles and vesicles, they are being formed inside the plants, and this ultimately provides the same function, performs the same function, providing the excess of nutrients as well as the moisture and the other growth factors which are essential for the growth of fungi. So that is regarding the mycorrhiza organisms. And again, one of the another one that we can look for is the endosymbiotic association or the endosymbiotic interactions. This is one of the typical type of association. It is something slightly different from symbiotic mutualism, that we will see that how the condition is being changed. It is the association between the Paramecium and the Chlorella group of organisms, Chlorella or the algae. And these algae or this Chlorella, they are present inside the cytoplasm. Large number of Chlorella, they are existing inside the cytoplasm of the Paramecium, and both the organisms, they mutually undergo for the symbiosis. Paramecium provides the protection, they allows the motility. They took this complete structure towards the sunlight so that the Chlorella can go for maximum photosynthesis and the fixation of carbon dioxide. Now in turn, the Chlorella which is there inside the cytoplasm will undergo for the fixation of carbon dioxide, will produce the starchy material, will produce the carbohydrates which are made being available to the Paramecium. But here the condition may be changed. Here the association is different in the sense that when the conditions are not providing the photosynthesis, in the absence of the light, this Paramecium may undergo for the digestion or the ingestion of the Chlorella which is there inside the cytoplasm. So in this way, the association is becoming totally different. Of course, we are saying that mutualistic association are symbiotic associations. They are providing the growth factors to each other. They are benefiting to each other. But here, friends, the association is changing, that when the conditions are changed, the interaction will be changed, and this positive association may become harmful, particularly to the Chlorella group. You can say that it is depending on the environmental conditions. Now the last one regarding the commensalism. See here, this commensalism is the association where two organisms will go for the interaction but one of the organism is benefited, the another organism may not be benefited, or even it is also not being harmed. The organism, you can say, these organisms, they are having a unidirectional type of symbiosis, where one organism is undergoing for the secretion of large number of different ingredients. The complex constituents like lignin and cellulose, they are being degraded into a simple form, and these simple forms, they are utilized by the other group of organisms which are existing in the soil. So this is again one of the very typical example. Then again you can look for some of the other large categories of the organisms which are producing the growth factors and vitamins in the surrounding environment, and again they are being consumed by the other group of organisms. Commensalism, in short, you can say that one of the group is acquiring the benefit, whereas the other group of organisms, they are not going for any benefit, but they are benefiting. They are in service of another group of microorganisms. So friends, here this is one of the very important aspects, that the combinations or the interactions, they are one of the important aspects for the survival of the life, for the maintenance of the ecology, stability of the life. If the interactions are not going on in the soil, then you can believe that the food chain or the nicks, they will be disturbed, and there will be existence, or there will be the presence of large number of only one particular type of organism. To control these organisms, the nature has developed this phenomena that they must go for different types of interactions. Human beings, as for example, if we are learning some lessons from this, then we may go for these type of mutualistic association. They may benefit to each other. Thank you very much. Now regarding the negative interactions, my partner, my colleague, Dr. B.V. Rawal, will go for negative interactions.
Well, soil is the outermost covering surface of the earth, and it is made up of organic and inorganic constituents. For soil microbiologist, soil is in fact a matrix